The Founders Memorial Award was established in 1958 to honor the memory of scientists who made outstanding contributions to entomology. I am ecstatically happy this year that the recipient of the Founders Award is Dr. Anthony James, a distinguished professor at the University of California at Irvine. His amazing record of accomplishments is far too lengthy to recount in this session, so you can find all the biographic information on page 25. But please, let me say he's honoring this. The honoree this year is Dr. Edward Knipling, and please help me welcome the remarkable and very impressive Dr. Anthony James.
Well, it's a real honor to be here. You can hear me, I hope. I've got a little thing so I can wander around a bit here. I appreciate the fact that May kept the introduction short. This isn't actually about me. This is about honoring one of the giants in our field. And I'm going to take the opportunity tonight to talk to you a little bit about him and then draw a somewhat personal art, personal in the sense of the field that I'm in, not me personally, with the work that he's done and the work that's going on in some of the fields that are ongoing today. Now, one of the challenges of doing this kind of work is, of course, that I'm not sitting on a slide deck of 100 slides, all right? This is all new content, all right? And to read someone's full output over the years is quite a challenge, and to select the things that you want to talk about and present means that you're bound to leave some stuff out. So thing number one is, for all of you who believe you should have your name in this and aren't, I apologize, we can add that later on. The second thing is, to what audience am I actually targeting my remarks? And I know if I'm talking to the people that know him, they're going to say, well, he missed this, he missed that. I know if I talk to his contemporaries or people of my age, we've all read about the papers and stuff. And so what can I say that's new to that? So I'm actually going to be talking to the students and younger scientists out there. And so these remarks are addressed to them. And for those of you who aren't in that class, you know, think wistfully about your youth. All right. So this is really an odd format, all right? I'm standing here in the middle of lots of things, and most of you probably want to look at me and the slides at the same time, but it's not about me. Once again, look at the side here. And there are a lot of open seats over here if you can't see enough. So we entitled the talk, or I entitled the talk, The Shoulders of Giants. And this refers, of course, to the famous saying that if we can see far, it's because we're standing on the shoulders of giants. And we're talking about Dr. Knipling and the dawn of genetic control. So I learned a little while back that when people give talks, they often are answering questions that haven't been asked. So I'm putting up here the questions at the top so you know what I'm talking about. I think it helps out a little bit. So what should we know about Dr. Knipling? First of all, he had a major role in discovering the development of sterile insect techniques. Most of us know about that, and we're going to talk a little bit about that and get into some of the data associated with that. But he also had a really interesting role in the development of DDT applications for controlling vector arthropods. Significantly enough, as you'll see, that he actually received awards for this. So there's two things you should know about him that are significant in his work. Another thing, he's definitely a man of the 20th century, and I don't mean that as a negative critique. You'll see how his work basically bridged major aspects that were going on in the 20th century. He was born in 1909, he died in 2000, and between that, he had education at Texas A&M as an undergraduate, did some work with USDA in Texas and USDA in Oregon before going to Iowa State to get his degrees, and then went to the USDA in Florida, and then had obviously major administrative roles in the USDA. And so we see his career stretching across this transformative generation. And along with that, in terms of this concept of a generation, of course, he was of that generation. They said the best of the generation. And this is an image of Dr. Knipling. There's actually no pointers here, so it's not. I anticipated having one, but we don't. So here. This is a picture of Dr. Knipling, Dr. Bushel, and Dr. Lindquist in 1943 here in the land of Florida. All right, and a conjunction effort with the U.S. Public Health Service to develop ways to control insects that would have impacts on the Allied troops during World War II. It's notable for two reasons. One is to bridge this gap between the USDA and the Public Health Service, which, in a sense, my being here is demonstrative of that. I'm not in the Public Health Service, but I work on public health problems. And also the fact that it was in Orlando, Florida. All right? So this happened here quite a while back, before most of us in the room were born, at least the ones I want to talk to. All right. And so he also was highly honored during his career, sometimes with dubious awards. He received the Golden Fleece Award, which was given out by Senator Proxmire for waste of money. And what earned him this achievement was a grant for the sex life of the screwworm, all right, which when Proxmire saw that, he thought, this is absurd, all right. To Proxmire's credit, he later recanted the award, and in fact, it doesn't show up in his official documents, as far as I can tell. Okay, and Dr. Knipling was honored very recently with the Golden Goose Award, also initiated by a congressman, but in this case to award something that had a lasting and major impact. Okay, so while we can talk and joke a little bit about the Golden Fleece Award, the Golden Goose Award awarded this year by the USDA ARS is significant. All right. And then underneath here, there's a list of awards that he's won. You can see the two that I was talking about in terms of his Public Health Service, USDA service. They're on your left, which is the Medal of Merit and the King's Medal for Service that were awarded by the English for the development of the technologies to control vectors that vectored human disease during World War II. You obviously got the National Member of Science as a member of the Academy and received another of other prizes. And this is the year that they're celebrating Dr. Knipling at the USDA, and this is the front page of the award for the Golden Goose. And there's a lot of materials online that I encourage you to see for those of you who are interested in going in a little more depth than we're able to do today. He was also plenary speaker at ICE in 1972. Our distinguished president sent me this, Dr. Berenbaum sent me this. And his opening address, which I guess I'm equivalent of right now, well, he was talking about entomology and management of man's environment. And then May told me that, well, he got kind of a mixed review on it. And I'm thinking, was she telling me that as an information thing or to put pressure on? Okay, so sweating a little more on that one. All right, so what's the problem? Well, as we know, there are a lot of beautiful, wonderful, and useful insects out there. However, many of these cause significant diseases in plants, animals, and humans. And basically, the challenge here was Dr. Knipling versus the screwworm Cochleomya omnivorax, all right? And here is the adversary here. It's a screwworm. It's a true dipteran. It's a diclifrid family. And it's a nasty bug, as we'll see, or actually nasty fly, as we'll see shortly. In the other corner here, we have Dr. Knipling, known his common name as Nip to his colleagues. He was with the USDA, and of course, we credit his parents as being the authors for there, but he's obviously a human being. All right. So what do we need to know about the screwworm? Well, it's a holometabolous higher order dipteran. It's an aggressive ectoparasite, all right? And we'll see, I decided I wasn't going to show you nasty images of what the screwworm could do. For those of you that are inclined, that generation of, vampires and all that kind of stuff, you can go look. They're nasty and they're ugly. But aggressive means this is an insect that doesn't just get on you and feed, sip a little blood like the ones that I work on. This one will eat you, all right, literally. And so look that up for those of you who are so inclined. It's before dinner here. But eggs are laid in wounds. The larvae hatch out, and they actually feed on the living tissue, all right? Unlike the flies that are used to treat gangrenous wounds and stuff who only eat the dead tissue, these actually eat the live tissue. And if you have enough flies, it's a problem. They cause the disease called myiasis, and it's in domestic and wild animals, and occasionally in humans. All right, so the economic impact before Dr. Knipling's work was significant. This is just data for American cattle ranchers, but we're talking about animal losses, a loss of stock, the monitoring, and then the actual treatment. And this was way back in the 50s, and it was about $200 million a year, which is equivalent to almost $2 billion a year now. And this is a pamphlet that was describing some of the efforts to control these before Dr. Knipling, which talks about the need to inspect your livestock, collect the larvae, treat, and spray. And you can see, if you can look at there, the regimen is actually quite laborious and highly expensive. The distribution of the New World screwworm was widespread in the Americas, from the temperate subtropical regions, southern United States, all the way down to southern South America. So what are some of the tools then that were nascent at the time that Dr. Knipling took advantage of? And you got to remember, he did this as a student. He was starting to think about this. So it was very early on. All right. Well, there was efforts in some tools that were available in insect ecology and behavior and in radiation biology. He was able to put these together in a very unique way. First, this is a picture of him with his colleague, Dr. Bushland. So they're called Nip and Bush, okay? I guess if you had a multisyllabic last name in those days that got abbreviated, I'm always going to be James. I mean, there's no way to shorten that. But it's what they observed, and actually Knipling observed, was that screwworms are present in relatively low densities. Despite the fact that the larvae, there are a lot of larvae that are laid into these wombs, the actual adults were present in low diversity. But then in something that actually was quite transcendent to many fields that we work in, the screwworm females only mate once during their lifetime. And this was key. And he thought a lot about how he could use this sort of biology and behavioral aspects to approach the problems that he was in. But running in a different track were people who were working on radiation biology. And I'm going to be giving timelines here, just here to give credit to people that contributed to these efforts that sort of coalesced in the work that he was doing. So Runner in 1960 realized that X-rays could actually induce sterility in insects, and I think this is what's called the cigarette beetle. And then H.J. Muller in 1927 had a series of publications that show that X-rays can induce sterility mutations, chromosomal rearrangements, et cetera, in the fruit fly Drosophila melanogaster. And for that, he received the Nobel Prize. All right. The reason this was a big deal is that previous to this, geneticists had to work on existing variation, existing materials. If they wanted to do crosses and look at genetic variation, they had to deal with what was already there. Now, it worked pretty well for the guy named Mendel, right? I mean, he had all the P's and everything, and he figured out a whole lot with existing variation. But geneticists were really eager to have tools that would allow them to make new mutations, new phenotypes, and things that they wanted to study. And Muller's work actually provided that. All right? It provided for the first time the opportunity to make new mutations at will and study them. Okay, so that was a big deal. When you think about it that way, yeah, he got the prize. It was worth it. All right, so what was the key connection? Well, in 1950, Muller wrote a paper, essentially a review article, talking about the application of radiation to genetics. And in it, he talked about the fact that the radiation, in this case X-rays, can generate lethal mutations in sterility. And Knipling saw the paper and the light went off. And he said, whoa, okay, I've got a way to approach this problem that I've been thinking about. This is in the 50s. He started thinking about it in 1927, at least as far as we can tell. So he'd been stewing on it for a while, trying to figure out how to make it work. And in 1955, he published a paper. Now, it's not very long, so I'm going to give you a minute to read it. No, I actually can't do that. But anyway, I just want to show you that this is the way science was done in those days. This is actually an article that appeared in Science, but it was based on 3 papers that were actually published in a journal that we should all know about, which is the Journal of Economic Entomology. All right, so this was in the old days when you could have all your data in one paper and then have a science paper that just was sort of the abstract, okay? We don't do it that way anymore, but some people still think things happen. But you look at the title here, Atomic Radiation for Insect Control, and you look at the date, 1955. So this is sort of contemporary when people were starting to make movies about Godzilla, all right, the ants in LA. For those of you, I said I was going to talk to the students, but now I'm reaching out to people in my generation. There's a great movie called Them about giant ants that ended up in the LA River Basin. But radiation caused that, all right? And there was a lot of fear of radiation at the time, but there was efforts to see if radiation could have peaceful applications. They talked about the peaceful atom. And so this is why this thing has atomic radiation for insect control. They got away from X-rays and started using gamma rays. Okay? But there were 3 papers that were in this journal that basically described the data that Dr. Knipling and his colleagues developed. And we'll go look at some of that now. So what was the first big result? Once again, his discovery that these insects are present in low density. His discovery that the females only mate once, and that if you radiate them, that you can cause sterility. All right, he thought, well, what he could do was, of course, irradiate males, have these males mate with females, and as a consequence of that, have non-productive matings. And as a consequence of that, drive the population to suppression. So he had a trial actually in Florida that didn't work so well because when they released the insects, they were able to get suppression, but they weren't able to create what's called a barrier area, and the flies would just come back in. Right? So they had an opportunity working with someone called Baumhauer to go to the island of Curacao. So they went to an island place first because they thought that the immigration or reintroduction of the pests would be limited by the fact that this island was relatively isolated. And so what they did is they sent up a number of goats and pins. There are 11 pins on the island that they had there. That's a color picture, so that's not likely the picture of the goats that they used, okay? This is a while back. All right, but you get the idea. You've got goats stuck there. It kind of reminds you of Jurassic Park, you know, that poor little goat waiting for the Tyrannosaurus to show up, right? In this particular case, it's a bunch of really nasty flies to the same end, if you think about it, all right? Nasty. All right, so once again, I didn't realize I wasn't going to have a pointer here, but basically this is a data table from the Baumhauer paper, and if you look at it, even if you squint, you can see that there's sort of 2 rows of data there. There's before release and after release. And before the release, if you look way over on the left in the yellow box, are the number of egg masses. So these are what would happen. The flies would land on wounds on these goats. They would wait a while, and they would go and they would investigate these goats and ask, okay, how many egg masses did I find there? And then they would try to hatch them out. All right? And so what you see there are the percent that are sterile. So before release, all of them were fertile in the sense that they hatched out larvae that could grow and eat the poor goat. Okay? But as they started to release, which is the group of data toward the bottom there, they started to pick up egg masses that would not hatch. Okay? And by the end of the experiment, 11 months later, not only did they get to the 100% of the egg masses could not hatch, they got to the point where they weren't even getting egg batches on these wounds. All right? This is a phenomenal success. 11 months for complete eradication of this fly. And that's actually an image of the larva. You can see those nasty little mouth parts there that facilitate its ability to feed. And as a consequence of that, then the release of these sterile insects through aerial releases was able to drive the screwworm, essentially eradicate it from the southern United States, push it through Mexico and down into the Isthmus of Panama, where they created a barrier that would prevent reintroduction. And now there's still efforts to maintain that barrier and push it into South America. So if we look at sort of a timeline here of the things that were happening, we can talk about the Runner's work early on, Muller's work, Knipling in '55, and then we have this large thing represented by a circle here, which is sterile insect technique. In addition to radiation, which is now currently gamma radiation, we also use, we meaning the field, use chemosterilization. All right, so what are the requirements of sterile insect technique? Knipling had 5 things that he suggested this would be useful for. There were requirements for that. Insects need to be reared economically in mass quantities. All right, it had to be cost effective to do that. They have to have a suitable life stage for dispersal, something that you could actually spread. They could be chemically or radiologically sterilized. So that had to be demonstrated. The insects only mate once, and it's amazing how many insects this is actually true of, all right? So this works to our advantage. And they must have low inherent population densities, because you've got to release a lot of these, and if you're trying to release more insects you're trying to release insects, more that are in your lab that are actually in the field, that can become quite problematic. And later on, thinking about this, we might think that we could add a couple of other things. No significant assorted mating, low natural dispersion, and monitoring systems are important. But the 5 things that he highlighted are significant and a good way to discriminate whether or not this technology could apply to this any insect that you're trying to target. And he suggested that this technology would be applicable for controlling pests that are present in small numbers, for ones that are newly established. So we can think of this in terms of invasive species. We can prevent invasive species for this. And as an adjunct to other control methods. And I like this last one because this is pretty much everything, right? If you can get it to work in conjunction with something else, that would be great. All right, so what are the legacies? So it was an interesting way to, I think, present, well, when you do something and you call it interesting, it sounds kind of weird, but I thought this might be an interesting way to present this, which is to look at the current activities here. And this is the latest report from the IAEA, Insect and Pest Control Group. This is an International Atomic Energy International Atomic Energy Agency, and they're still using atomic energy, as we said, atomic radiation on that 1955 poster, or paper, to work with insects here. And the legacy, and this is just from this one report alone, we have 2 species of Lepidoptera, we have 3 species of mosquitoes, we have a bunch of fruit flies, and I know that Drosophila in there is an honorary fruit fly, but we can put that in the group there. The tsetse, stable fly, and even a beetle, all right, that are being considered. So these are, just in this journal alone, reports on the use of this technology. So this legacy is robust, all right, and continually continues to this day. People are still working with it. All right, so this is sort of the thread that he had that was going along. So what are the kinds of things types of stuff that I'm interested in that were in parallel? So we had vector biologists thinking about malaria eradication, all right? And they were thinking about, well, we can go through these steps where we can control malaria, eliminate it, and if we can eliminate it everywhere, we can have a global eradication. What should you know about malaria? It's an infectious disease, an intracellular protozoan parasite. The image on your left are human red blood cells. Those little purple spots are parasites. Those cells are supposed to be empty. When you see those parasites in there, you have a problem. It's transmitted by mosquitoes of the genus Anopheles. You see there in the picture the characteristic profile of a female Anopheles feeding. She's got that sort of 45-degree attack angle. She lands that way like she's ready to go. And one of the things that's really interesting about this, there are a lot of different malaria parasites, and they have a high degree of host specificity. So there are human parasites, there are lizard parasites, we have some guinea fowl here, and rodents. They have their own types of parasites. Humans don't get mouse malaria, mice don't get human malaria. There's a lot of diversity of the parasite out there. There are no free living forms of the parasite. They're either in the vertebrate host or in the mosquito, okay? And in 2014, we're waiting for the updated data for 2015, but there were somewhere near 250 million cases and about 600,000 deaths as a result, in humans as a result of malaria. Well, what should we know about mosquitoes? Because we're at the ESA, holometabolous insect, they're true flies. Only adult females feed on blood or are involved in transmission, and blood is a source of protein for nutrition and egg production. All right. Now, the work of Knipling and his colleagues stimulated a lot of the vector biologists to think about what could possibly how this could possibly be useful in the work that they were doing. And they began to look in their own fields for significant early events, early discoveries that might inform the way they approach things. All right. And they were able, as a consequence, to develop tools that could achieve local control and elimination. We see the use of bed nets, indoor spraying, environmental modification, and drugs. Those are the images that are across there. But then if those things work so well, why is malaria still here? Well, some of these tools have never been applied for geographical or political or economic reasons. Some of the tools, they just don't work, all right? Bed nets, for example, work on mosquitoes that feed at night, and it turns out that when you suppress them, you still have malaria, and you realize, oh, there's some mosquitoes feeding in the daytime, all right? So you need a technology that will go after them. And then we have circumstances which many of us face, which is that the tools that used to work no longer work, and the big one here is insecticide resistance. All right, so people were thinking about the work that Knipling was doing and thinking about, what about this genetics? You know, we've been using chemistry for drugs, immunology. Our colleagues have been making a lot of effort. Dan Strickland's here. We've a lot of effort on vaccines, ecology and environmental management, behavioral sciences to try to suppress malaria. The question is, all right, is there something in genetics that could potentially be useful for this? So what are the genetic outcomes? Well, a genetic outcome would be population suppression, a sort of a genetic analog to an insecticide, a technology that would reduce populations to the point where you don't get bit or you don't get bit, essentially, you don't become infected. But then there's an alternative, which is what was called population replacement, modification, alteration. It's got a bunch of names now, unfortunately. But here you change the genetic ability of the mosquitoes to transmit the parasites, recognizing that while the mosquitoes are problematic, nobody likes to get bit, the thing that causes disease is actually the real target, the malaria parasite. So these genetic outcomes could actually, in addition to targeting mosquito for population suppression, you can consider a different approach that would go after the parasite. So what are some of the tools then are being applied to these types of approaches? Well, we have genetics, and we'll review that quickly. We have now molecular genetics, and within molecular genetics, we have things like transgenesis and gene drive that are being made available to us that allow us to get at some of these technologies. So let's look quickly at some of the advances in population suppression. Way back in the 30s, Vanderplank was looking at hybrid sterility in the tsetse fly. Basically, the idea that if you had two sibling species cross-mate with one another, that you would get sterile hybrids out. So you could use one sibling species to drive the other one to extinction. Cerebrowski came up with models of negative heterosis and underdominance, ways to engineer chromosomes in a way that would give population suppression. Hickey and Craig described Mitotic Drive in Aedes aegypti. Craig was honored a few years back in a founder's lecture here. But basically, they had a meiotic drive system, a natural meiotic drive system that drove the population to all males. And if you have males only, it's a pretty sad population. It will crash. People looked at translocations and cytoplasmic incompatibility. Ed Davis was involved in a lot of this work. And Alfie, who's in the crowd here, and Max Scott, who may be here somewhere, looked at conditional dominant lethals in the 2000s. And the consequence of this, we have a lot of tools for population suppression. And this is a table from a paper by Clausen and Curtis here that lists some of the efforts in mosquitoes that were actually able to achieve population suppression. Once again, this is just to show you that there's information out there. But you can see 3 lines that are heavily enhanced. Culex quincunfasciatus, they actually got eradication in a small village. Anopheles albumensis, 100% sterility in a field, and another place of albumensis, natural population suppression. So these types of technologies, existing technologies, can use to suppress mosquito populations. And currently, Oxitec, a company that's developed conditional dominant lethals, release of insects carrying dominant lethals, has released trials in the Cayman Islands, Brazil, and others targeting Aedes aegypti. So that's sort of the population suppression thing, and we'll finish off the last 2 minutes here talking about the population alteration or modification. So all the while, when people were looking at the malaria problem, they realized that there are places where they have Anopheles mosquitoes and they don't have malaria. So just having Anopheles mosquitoes didn't necessarily mean you're going to have malaria. And they actually gave it this name, Anophelism without malaria, all right? And they were perplexed by it. And it turned out that in many cases, it's because the Anopheles species were not able to support the development of the malaria parasites. Okay? And so this led to this concept of vector competence, and this is an intrinsic ability, a genetic ability of a particular vector to support the development of a pathogen. And we know that, in a very simple way, we know that there's got to be something genetic there, because there are a lot of blood-borne pathogens. We can talk about malaria, leishmaniasis, HIV, hepatitis, and there are a lot of things that feed on blood. We can think about mosquitoes, ticks, fleas, et cetera. And not everything that feeds on blood transmits all those pathogens, all right? And we see a clear association of specific pathogens with specific blood-feeding arthropods, and there's a genetic component to that. So we can start to tease that apart and ask, all right, what's important about that? And what we find is a lot of really interesting biology associated with that. There's going to be the great talk later on in the session here, plenary talk, about the immunology of that, and then about the intrinsic biology of a parasitic organism migrating through an insect. So there's a lot of interesting stuff that comes out of that. But there's a genetic basis for that. That is, genes are involved, gene products will actually have an impact on whether or not a specific pathogen can develop in a blood-feeding host. So way back in 1929, Clay Huff was able to select for resistance to avian malaria in mosquitoes. He was able to grow populations of mosquitoes that could transmit, and then through selection procedures, get 2 populations, one that could and one that couldn't, indicating that there was an underlying genetic basis for that. McDonald did the same thing later on, looking for resistance to filarial worms. Craig, which you can see why he should be honored on the podium here, was also mapped resistance for malaria. And then in 1986, Frank Collins, working with a number of people in NIH, was able to do the same thing for human malaria in Anopheles species. So the idea is you were able to select for variants that had resistance. Once again, starting to probe this genetic aspect of it. So in this context, there's one other person I would like to introduce you to, who is Chris Curtis. And Chris Curtis was a biologist at the London School of Tropical Medicine, and he thought a lot about the same sorts of genetic approaches to preventing the transmission of parasitic diseases. And he came up with this idea of population replacement, and which is now was called modification for a long time, and now it's called alteration. And once again, there's a seminal paper in 1968 where he talked about the fact that you could think of mutant genes that the presence of which in an insect population would prevent that population from doing something, in this case, transmitting malaria. And he said at fixation, this population then would be harmless to man, and this type of procedure might be preferable to methods that eradicate this pest species because it doesn't leave an empty ecological niche. All right? And so the idea here was that one could do this change of a population from one type to another. And so there have been a lot of us who have followed up on these ideas. I knew Chris Curtis, knew him quite well, and I think he probably thought I was kind of a Frankenstein. He had this great idea, and then a bunch of molecular biologists took it and went crazy. But that's what happens. And so the question then is, what are the applications of molecular biology and genomics to mosquito population suppression and alteration? And we've got the first protein coding gene in 1989, RFLP maps, genetically engineered pathogen resistance, transgenesis systems are available, a first pathogen resistance line, first mosquito genome sequence came along, first gene drive system in 2011, and very recently, the use of the CRISPR-Cas9 technology. So these are all new technologies that can be brought to bear on these particular problems. Now, this talk was to draw an arc between Knipling's work and the modern genetic approaches to trying to prevent the transmission of vector-borne diseases. I will acknowledge in this box here that there are non-genetic approaches, okay? We don't go into them because the talk isn't about that, but I will like to acknowledge biological control agents. We have entomopathogenic fungi, toxic bacteria, Wolbachia. We'll hear there are a lot of talks about Wolbachia at the session here, and then paratransgenesis. All right, so take-home messages. I think I'm on time. If that's the case, it's just water. In California, that's important, though. We salute people with water in California. Beer's cheap. All right, so the take-home message, once again, talking to the students. It's great to have novel ideas. We want to encourage this, all right? A lot of times you're in training and you learn about these people, you learn about Knipling. Some of you might even read the papers that I've published and stuff. And you can't have your thinking confined by that, all right? We like our ideas, okay? Some have done done better than others, okay? But we really need new ideas, all right? We need creative new thinking, all right? And Knipling was the embodiment of that. Once again, as a student, he started to think about this. Okay, it took him a little while to play it out, but he had a war to fight in between, all right? But the ideas are important, all right? The second thing is the multidisciplinary approach, extremely important. Okay, so go to that weird-sounding seminar. Even if you only heard, if you don't understand most of it. I mean, Knipling knew nothing about radiation biology. He wasn't a radiation biologist. But he saw that paper, and the light, I use the image, said twice, the light went off. Right, he saw an application for something completely different outside the area that he was in. And he made the connection. So it's really important to have this sort of multidisciplinary approach. All right? I like reading about ants. I'd love to be able to do something about this invasive Argentine ant. It sounds like it's going to be a real problem. But I'm paying attention to it because maybe some point something will click. Maybe not. Who knows? All right, but this multidisciplinary approach is important. And then finally, what Knipling did for many, many, many of us were still waiting for our stuff to work, all right, was he was able to do it, all right? And so whenever we get criticism, we go, well, but Knipling was able to do it, all right? So we figured that that's an impetus for us to keep on working. It took him a while. He was ridiculed, as you saw, with the Golden Fleece Award. In fact, early on, people didn't believe it before he even got the Golden Fleece Award. People did not believe that his work was going to play out, but it did. All right, so he had a great idea, a multidisciplinary approach, and he actually achieved something that has a long-lasting and significant impact. Now, I'm not about to claim that I will ever have that, and I can't promise that any of you will ever have that, but what I can say is if you don't do it, it won't happen. All right, so I'm urging all of the younger students or younger scientists and students in here, if you have I'm not going to call it a crazy idea, but if you have an idea where you can connect the dots and you think you can make it work, it's worth putting in a life's worth of effort. And that's what Knipling has done, and I think that's the major thing he's taught all of us who don't specifically work on the screwworm. Thank you very much. But yeah, I guess it's time for Q&A. I love it. Just Q. Oh. Oh, you get to ask questions and I don't get to say anything. Anybody have anything they want that's burning, kind of hard to see here? There we go. I guess not. All right.
No question?
No.
Then let me give you your award.
Oh. I can hear you. Do you think? Say,
Does this country have the moral will to undertake something similar to
excellent question. Does this country have the moral will to undertake something similar to what Dr. Knipling has done?
That's a question.
The debate is tomorrow. I think we have the moral will. All right. They often say you get the leaders you deserve. All right. I don't believe that. All right. How we could possibly be sitting. You have to turn the cameras off now. All right. You got me wound up, and I thank you. How we could possibly be sitting and fighting about not funding money to fight Zika. I just can't understand that. So that's not a moral issue. Well, maybe it is. All right, but for those of us who work in the area, we have the will. We know that people believe in the types of, not when I say we, I mean us, all right, the entomological types, that people believe in our work, and I just hope they catch up to us. And maybe the point of doing a lecture to this is not just to talk to you, but to get this information out once again to the people who control the money for this kind of stuff. You know, the USDA ARS honored them this year. Hopefully that'll put some pressure on it. I'm an optimist because I'm a scientist, okay? So all I can tell you is I sure hope so. I really do. Great question, though. Yes, please. Oh, wait, there was one more question, yeah.
Really quickly.
Now we're having fun.
Dr. Knipling developed a system that caused the eradication of a native insect pest, a screwworm, right? Mosquitoes are a keystone species in a lot of ecosystems. Do you see a dilemma in developing something that might eradicate a species at Salem malaria?
Okay, so you and I could have, so the question was in circumstances where mosquitoes are a keystone species in a lot of ecosystems, do I see a dilemma with eradicating them? So I would say that there aren't really that many demonstrated cases where mosquitoes are actually a keystone species, okay, but we can have that conversation later on. But I will tell you that the places where they're the most problematic are places where they're invasive. All right, so I just came back from the International Union of Conservation of Nature in Hawaii, working with the people who are concerned about preserving the birds in Hawaii. All right, there were no mosquitoes in Hawaii until 1830s. All right, it's an introduced species there. It's devastated bird populations below a certain altitude. All right, avian malaria, avian pox have done that. I had as scientists, we tend to be detached, we're seeking the truth. But I had a very emotional experience. No, I wasn't emotional. Well, I was actually. I'm telling you about it. talking to a young scientist working for the San Diego Zoo. And he's charged with preserving a bird species of which there are about 200 members left. All right. He has about 40 of them in captivity. And what he's doing is he's breeding them in captivity to maintain the species. He can't let them go, though, because if he lets them go, they go out and they get malaria and they die. And he came up to me and he was talking to me and he said, for the first time, he's a young guy, and he doesn't want to lose a species on his watch. All right? And he came up to me and he said, for the first time, we have a chance to do something about this. I mean, you know, he was just, you know, it's like those of us collected insects and put them in boxes and kept them in our room, you know, our ant colony. I mean, he's handling these birds in that same sort of way, all right? And so our technology, at least to him, ours in the broader sense, have offered a possible solution to the loss of this bird population. In fact, 30 other species that will be lost in Hawaii in the next 10 years, all right? And it involves eradicating mosquitoes from Hawaii. I think that that's probably a worthy goal.
And in 5, yeah, round of applause. Just a reminder, in 5 minutes, the doors open for the International Congress of Entomology. So before they do, let me present you.
Let's not let them in.
Yeah, with your prize or your gift.
It's heavy.
Yeah, and I think
They
are
want
there
to take a
pictures
picture?
being taken?
Yeah, let's go. There's an extra, so we got to stand. All right.